Hello, everyone. I'm Jun. I'm a assistant professor and lab head of single-cell genomics and population dynamics from the Rockefeller University. Thank you for giving me this chance to introduce our previous work about tracking development and the cellular level. So as we know that we all develop from this tiny single cell, and this tiny single cell continue to divide and differentiate into hundreds of different cell types, and they form different tissues, organs, and finally our body. So we are interested to understand how these different cell types are generated in development. So you can imagine to answer this question, we need to comprehensively characterize this different cell state and their dynamics during development. So traditionally, people study this question by, for example, imaging techniques. In this case, people use some cell type specific protein markers to label these different cell types. But you can imagine here, for one or two different markers, actually it's limited to characterize these hundreds of different cell types in a single experiment. So alternatively, people use another technique called single-cell genomics, or most by single-cell RNA sequencing techniques, to characterize these different cells, not based on one or two markers, but based on hundreds to thousands of gene expression. So in this case, we can comprehensively characterize different cell states with less bias, but there's also some limitations for single-cell genomics, and the limitation is the throughput. For example, here we look at the cell number in the neuron system from different organisms. Here we have hundreds to millions, even billions of cells, right? Like in the neuron system. However, the throughput of the single-cell genomics, like when I just started my graduate school in 2015, it's just a couple thousand cells. So this is very low compared with this millions to single cell like number in real tissues. And this makes it very challenging to characterize some rare cell types or to study cell population changes during development. So when I started my graduate school in Dr. Jason Jarrett's lab at University of Washington, my main aim is to develop a high throughput single cell RNA sequencing technique. And the key is the combinatorial indexing. So combinatorial indexing is a strategy that enables single cell detection without isolating single cells. And here is the trick. For example, here we start with a group of cells. First, we can split the cells into a multiple well plate. And for each well, we have like several thousand cells. And we can barcode the molecules in each cell with some well-specific barcode, right? And then we can pull the cell together. So each cell gets one barcode, but some cells actually share the similar barcode because it came from the same well. So the trick here is that we are going to repeat this whole process. So you can imagine after several rounds of random distribution, indexing, and pooling process, the molecules in each cell will be labeled with a specific array or barcode. And because all the molecules from the same cell, they travel through the same set of wells, right, in each round, which means that the molecule from the same cell, they will receive the same barcode array. So which means that if we lysis all the cells, extract all the molecules for sequencing, we can group molecules from the same cell based on this barcode array. In this case, we can identify single cell information without isolating single cells. So the key feature from this technique, you can imagine like the throughput of the technique depends on the barcode, like complexity or the diversity, right, that can be generated in this experiment, which means that the detection capacity or how many cells we can profile in a single experiment depends on how many rounds of indexing we do. For example, with just a 2-round indexing, here we can profile around 40,000 cells. And based on this, I developed a series of single-cell genomic techniques to profile single-cell transcriptome, single-cell both transcriptome and epigenome, or transcriptome and newly synthesized RNA from the same cell. And if we just include another round indexing with 3-round indexing, here we can profile like million cells in a single experiment, and we can use this to study the cell population dynamics in early development from both mouse and human. So here I just want to give you one example about how we use these techniques to study cell population dynamics in early development. So for example, here, if we look at the mouse embryos, like across the key stages of the organogenesis, from the 9.5 day to 13.5 day, they have like hundreds of thousands to millions of cells in each individual. And this makes it very challenging for conventional techniques to study the cell population changes and the whole organism scale. So using this combinatorial netting strategy, here we developed the SciLifeLab and use this to profile 2 million cells from this system across different developmental stages. And we can use unsupervised machine learning to group cells to different clusters based on their transcriptome similarity. So for example, by the cluster analysis, we can identify main cell types from mouse embryos that includes connective tissue progenitor to some very rare cell types, for example, the lens, the melanocyte, and other cells. So this represents the main cell state in mouse embryonic development. So the question is that for these different cell types, how the dynamic change or how they are generated in early development, right? Like from early to late developmental stages. So to track the cell state changes in this developmental time window, here we use another algorithm called UMAP that can project the related cell type into a developmental trajectory. For example, this is the cells colored by the main cell types as shown in the previous plot. And by UMAP analysis, we can see the cells like from the related cell lineage are grouped together. For example, this group of cells are all mesenchymal cells that include chondrocyte, myocyte, and other like, for example, fibroblast cells. And if we see this group of cells includes the neurons from the CNS system, includes the early progenitors and the differentiation neurons. So after this transformation, we can see the main cell types are grouped into 10 major trajectories, from mesenchymal trajectory to epithelial trajectory to different neural crest trajectory to endothelial trajectory. So because each trajectory here still contains like hundreds of thousand cells. For example, the epithelial cell trajectory is a small cluster here. But if you zoom into this trajectory, for example, here if we took out the epithelial trajectory and redo the same analysis, now we can see the epithelial cells can group into more complex 3D structures. And here the cells are colored by the main epithelial cell type based on their transcriptome signatures. And we can also color the cells by the developmental stage each cell comes from. So combining both information, we can use this to annotate the differentiation trajectory that form the epithelial cells in different organs. For example, for the mid-gut, hindgut trajectory, the foregut trajectory, which already separated into two directions, one to the stomach, one to the lung. We can also identify the epithelial cell trajectory that form different sensory organs, the kidney, and others. For example, here if we look at this trajectory, the apical ectodermal rich cell trajectory is a cell trajectory that involves in or digit formation. So if we took out this trajectory, redo the similar analysis, we can directly characterize the cell's transition from early developmental stage to the late developmental stage. And we can also characterize hundreds of genes that are differentially activated or suppressed like during the cell differentiation process. For example, here, if we look at the downregulated genes, we can identify some genes involved in, for example, proliferation regulation, like downregulated in this cell type. And this is also consistent with the fact that this cell type is a transient cell type in development. So this is just one example to show you that we can use this data to study gene dynamics and use this to study how the cell differentiate from early to late developmental stage. And this is just for a very small cell trajectory, right? So we use a similar strategy to profile, like to analyze other main cell trajectories. And in total, we can identify 56 cell trajectories that corresponds to the cell differentiation path in different organs and systems. Because this is single cell RNA-seq data, so we can also use this to study the gene dynamics or transcription factor dynamics to understand how the different cell types are developed in this critical time window. So in summary here, I show you by this high throughput single cell genomics, we can use this to characterize the cell population from this entire organism, and also use this to understand how the different cell types are generated in early organogenesis. And we also use this technique to profile the cell population dynamics in the later stage, for example, in the different human fetal organs. So because time is limited, so here I just want to show you one result. So from the mouse embryonic development, we identify 56 embryonic cell trajectories. And from the human, we identify 77 fetal cell trajectories. If we integrate these two data sets together, for example, in this plot, each dot represents a cell from mouse embryo, so human, and they are colored by the developmental stage. For example, here we can directly see the cell transition from the very early embryonic stage, like the 9.5 day. They transit to some later stage, like 10.5 day to 13.5 day, and then they smooth transit to the human fetal stage. So suggesting that using this data, we can use this to study the cell differentiation path, not in organogenesis stage, but also use this to infer the cell differentiation path from the early embryonic stage to the fetal stage. So at last, I want to thank the great support from my PhD mentor, Dr. Jay Junyue, and also the help from all our collaborators. Also, we are a new lab at the Rockefeller University. I want to thank the support from our current lab members. Also, I want to thank the Science and the SciLifeLab committee, for recognizing our work, and hope everyone can have a great time in the ceremony. Thank you.